Okay, so the title is Quantitative Systems Toxicology Modeling Provides Novel Mechanistic Insight into Disease-Related Tolvaptan Hepatotoxicity. And it's my pleasure, if you advance, this just says what our disclosures are. I no longer have equity interest in DILIsym services or Simulations Plus. Next. And the modeling that you're going to hear about from Jim is with the drug called Tolvaptan, which is approved for the treatment of autosomal dominant polycystic kidney disease, or ADPKD. However, it comes with this black box warning of serious liver injury. And two things. One is this was a surprise in clinical trials because Tolvaptan had been on the market for years to treat hyponatremia, and never in the clinical trials leading up to approval for that indication or post-marketing had liver safety been a concern. So I think it's clear there's something about this disease population that creates this susceptibility. The second thing, if you look at the underlined statement here, they're recommending that liver chemistries be monitored monthly for a year and a half, 18 months. That's very unusual. If someone's going to develop a liver reaction to a drug, they usually do it in the first 6 months and certainly within the first year. And on the next two slides, if you advance, Jim, I'll show you two things that I'll come back to that I think the modeling has provided insight into. So what you see along the x-axis are the months in the study, the patients receiving placebo or Tolvaptan, active drug to comparator to placebo, 2-to-1. And you can see along the y-axis the proportion of subjects experiencing elevation in liver chemistries indicative of liver injury. And you can see that starting around 3 months or so, there's clearly a deviation. There's a on treatment emergent of liver toxicity. And the interesting thing is that it does continue beyond 12 weeks. And of course, the numbers fall off after that. I'll come back to address these arrows here. And if you go to the next slide, Jim, this is a patient that was in the approval clinical trials with ADPKD. And what's shown again along the y-axis is the time this patient was receiving drug, which is shown as a shaded area. And then the various liver chemistries, I won't go into the details, are shown along the y-axis as a fold upper limits of normal. And you can see it was really around 5 months when this injury started to take off. The drug was stopped, but in spite of stopping drug, really for weeks and a few months, the liver injury progressed, even after the drug was gone, only to improve. And then because this was unexpected, they went ahead and started the drug again. And then immediately there was elevation, there was sign of liver injury. And this we're now recognizing, and in many instances, we now can comfortably attribute this to an adaptive immune attack. There's something about the drug interacting with the liver that makes the immune system thinks it's virally infected, or in any event, there's a disease and they have to kill those cells. And then the prompt recurrence is immune memory. And I'll come back to this because I think the modeling that Jim has done really provides some different or novel insight into that. So you got a drug exposure, you get an adaptive immune attack, but in between, we believe there are a series of necessary but not sufficient steps, including a neoantigen, release of what are called danger signals, local inflammation. And this is the sweet spot currently for DILIsym modeling and what Jim will be addressing. So take it away, Jim.
All right, thank you, Dr. Watkins. All right, so you may wonder, is it Tolvaptan the parent or perhaps one of the Tolvaptan metabolites that may be leading to this Tolvaptan-associated hepatotoxicity in this patient population? Well, let's talk a little bit more about Tolvaptan metabolism. It's primarily mediated by CYP3A4. More than 20 metabolites of Tolvaptan have been identified. The two major ones are the ones you see here on the far right, DM4107 and DM4103. Because they're the major metabolites, these two, as well as the parent Tolvaptan, have been tested in the lab as inhibitors of hepatic bile acid transporters, as you can see on the left, as well as in the seahorse assay as inhibitors of mitochondrial function. While this one metabolite, 4107, had very little impact on hepatic bile acid transporter inhibition or on mitochondrial function, you can see that Tolvaptan inhibited at least 2 bile acid transporters and impaired mitochondrial function, but especially this metabolite 4103 inhibited multiple bile acid transporters and impaired mitochondrial function. So previously, DILIsym was used to model tolvaptan. So for those that are not too familiar with DILIsym, although I'm sure you are after attending some of the presentations during this conference, DILIsym is a quantitative systems toxicology modeling platform created by the DILIsym initiative. Multiple species are represented. Inter-individual variability is represented. Multiple liver zones are included in the model. And as you can see in the the diagram on the right, multiple submodels make up DILIsym, including, for example, a PBPK or drug metabolism and distribution submodel, a bile acid homeostasis submodel, mitochondrial function, or, for example, clinical biomarkers. So DILIsym predicts drug-induced liver injury via the intersection between PK or exposure, DILI mechanisms, biochemistry, and interpatient variability. So in this previous work, back in 2017 by Woodhead et al., what they did was basically first develop a model for tolvaptan and take into account this bile acid transporter inhibition and mitochondrial impairment due to tolvaptan and this one metabolite. Basically, the nice thing in DILIsym that you can do is turn off individual mechanisms and evaluate whether a specific chemical species or a mechanism is a primary driver of toxicity. So you can see that when all mechanisms are on, 18 out of 229 individuals from a virtual population experience ALT elevations. Turning off either tolvaptan or the bile acid transporter inhibition mechanisms, you can see that all the ALT elevations dropped, indicating that both tolvaptan and the bile acid transporter inhibition are key drivers of the toxicity. Turning off the metabolite, it was also shown that this was an important contributor to toxicity, since only still 5 individuals remained, but 13 individuals out of the original 18 had no longer ALT elevations. So back in my time at UNC, where I finished my dissertation last year, we gained some new insights into tolvaptan, hepatobiliary disposition, and potential roles of hepatic transporters, specifically using a rodent model of this disease called the polycystic kidney rat. And we saw that there's a role for reduced biliary efflux of this metabolite, and we saw dysfunction of a biliary efflux transporter called MRP2. So a little bit more about this MRP2 biliary efflux transporter highlighted here in the liver image in the center, highlighted with a red circle. It's a biliary efflux transporter expressed on the canalicular membrane of hepatocytes, primarily responsible for translocating organic and ionic compounds into bile. And if you look at DM4103, you can see that this carboxylic acetyl actually becomes negatively charged at physiological pH, which actually makes this compound a very likely candidate to be a substrate for MRP2. So we did some additional work in the lab, for example, using isolated perfused livers of polycystic kidney rats and wild-type rats. We perfused tolvaptan through the portal vein and collected bile and outflow perfusate samples. Using mass balance and PK modeling, we actually saw that there was a significant decrease in DM4103 in the bile of these PCK rat livers. In addition, doing Western blot for MRP2, in addition to other transporters in these PCK rat livers and wild-type rat livers, we saw a 3-fold decrease in MRP2 expression. We actually did some additional isolated perfused liver work with a MRP2 probe substrate and did some PK modeling, which found a 28-fold reduction in the biliary excretion rate constant for this MRP2 probe substrate, which is not shown here, but you can review that in the publication below. So with these new insights about polycystic kidney disease, of course, focused on the rodent model, but we wanted to use DILIsym to answer this academic question. Considering these new disease-related insights, so focused on MRP2 dysfunction and reduced biliary efflux of this metabolite, what is the impact of reduced biliary efflux on tolvaptan-associated hepatotoxicity? So for the DILIsym methods for this UNC-based project, the primary parameters of interest, as you may imagine, is the biliary excretion Vmax for this metabolite, since we're assuming it's a good candidate substrate for MRP2, and we see this MRP2 downregulation, and we see in the isolated perfused liver reduced biliary excretion of this metabolite. As a comparator, we also looked at tolvaptan's biliary excretion. So we performed various types of simulations using, for example, the baseline human in DILIsym. This was basically a sensitivity analysis where both of these Vmax parameters were modified. In addition, some virtual population, or SimPops, as it is called in DILIsym, simulations were performed in which a healthy volunteer virtual population consisting of 285 individuals with variability in 44 parameters applied to multiple submodels, specifically PK, bile acid homeostasis, and mitochondrial function was applied. And on top of this variability in these 44 parameters, I applied this 3-fold or 28-fold reduction in these Vmaxes to reflect those reductions we found in the polycystic kidney rat based on Western blot as well as functional data. So for the next few slides, I'll be showing you some selected outputs from the DILIsym modeling. Just to orient you on the layout of the following slides, I just want to introduce you the way it's set up. So on the far left, you'll see the sensitivity analysis in the baseline human, where the biliary excretion Vmax of the parent, so tolvaptan, was decreased. In the middle, you'll see the excretion, biliary excretion Vmax for the metabolite being decreased. We know that tolvaptan is a substrate for MDR1 or PGP. So this basically reflects like a reduction in PGP function, while the middle panel will basically reflect a impairment in MRP2. And we also explored the simultaneous impairment of both of these pathways. So we'll look at the default Vmax value as well as 6 log2 values below. So let's focus on the biliary tolvaptan amounts. So if you focus your attention to the left panel, you will probably notice that this is actually a positive control for this particular sensitivity analysis. We're decreasing here the biliary excretion of tolvaptan, and what we see is a decrease in the biliary tolvaptan amount, just like you would expect in this scenario, while there's no change in, for example, the middle panel. Some additional characteristics I want to point out here. So the vertical dotted line indicates the end of the dosing regimen. The simulation time was a little longer to potentially capture some return to baseline that we want to capture as well. And you may notice on the far right, where both the biliary excretion Vmax of the parent as well as the metabolite was decreased. You see some colored simulation lines not making the end of the simulation run, basically indicating a fatal event. So whenever you see these premature simulation terminations, you can call them that way, you basically have a very fatal toxic event, which you see in the far right, a few examples of. So even though, so what we're looking at here is the hepatic tolvaptan concentration. So by decreasing the biliary excretion of tolvaptan, we actually do not see an increase in tolvaptan liver concentrations. While for the middle panel, you can actually see that towards the end of the simulation, there's so much toxicity happening that the apparent concentration of tolvaptan in the liver increases. And this is also the case on the far right. And you can appreciate that there's this purple simulation run in the far right that actually did not make the end of the simulation that was able to complete the simulation in the middle panel. So there's even more toxicity happening on the far right than in the middle panel. So moving on to the biliary metabolite amounts, DM4103. In this case, the middle panel would serve as a positive control for this type of sensitivity analysis. By decreasing the DM4103 biliary excretion, what do we see? We see a decrease in the biliary DM4103 amounts. If you look at the far left, decreasing the tolvaptan biliary excretion, you actually get a slight increase in the biliary metabolite amounts due to some of the tolvaptan not being able to be excreted into bile, hence more metabolite being formed and subsequently excreted into bile, which is what you see in the far left. So decreasing the biliary excretion of the metabolite, as you can appreciate in the middle panel will lead to simulated increases in hepatic metabolite concentrations. So I know there's a lot of data here, so I would like to summarize the previous 4 slides in this table here with the different colored arrows indicating very undesirable, undesirable, or neutral or trends in the outputs I've just shown you, which are focused on the PK. So especially when the metabolite biliary excretion is altered, we see some very undesirable elevations in hepatic tolvaptan concentrations or the metabolite. Looking at some additional outputs, for example, the hepatic bile acid concentrations, we see in all of these 3 panels that whenever tolvaptan is administered, we see this initial increase in the liver total bile acid concentrations. And this is because tolvaptan and this metabolite that are now both present in this system are, as I mentioned earlier, bile acid efflux transporter inhibitors, leading to some bile acid buildup. In the case of the middle and right panel, you can see that eventually this leads to a gradual buildup of even more bile acids, and which eventually leads to this hepatotoxicity. Looking at the hepatic electron transport chain activity, so again, when tolvaptan and this metabolite is on board, initially we see this initial increase in each of these 3 panels. And this is because these bile acids that accumulate are actually they actually inhibit ATP synthesis, which leads to this adaptive increase in electron transport chain activity. However, while this metabolite is being accumulated in the liver, eventually this potent ETC inhibitor that the metabolite is basically counteracts this adaptive increase until this ETC activity goes down until the hepatotoxicity occurs. And this goes hand in hand with the hepatic ATP concentrations, where these basically drop down very rapidly eventually for the case where the metabolite is building up inside the hepatocyte. So again, a lot of outputs that I would like to summarize in this table. So again, when this metabolite's biliary excretion is impaired, we see these very undesirable elevations in hepatic bile acid concentrations, decreases in the ETC activity, and decreases in the ATP concentrations. As you can imagine, DILIsym has hundreds of additional outputs we could explore, but there are just 2 additional ones I just wanted to summarize in this table, which are 2 important clinical biomarkers, the plasma ALT and plasma total bilirubin, which, as you may expect, also very undesirably increase in the case when the biliary excretion of the metabolite is decreased. So what I've shown you so far were these sensitivity analyses in the baseline human. So we went on to do some additional simulations in the virtual population, or the SimPops, consisting of 285 individuals, on top of which we applied this 3-fold or 28-fold reduction in these biliary efflux pathways. So when none of these pathways were altered and tolvaptan was still administered at the highest dose, we still had 1 out of the 285 individuals, or 0.35% of the whole SimPops, that had an ALT elevation. The same individual had bilirubin elevations and basically satisfied the characteristics of a Hy's law case. When tolvaptan's biliary excretion was impaired 3-fold or 28-fold, very little changed. Only when the impairment was 28-fold, we see 1 additional individual from the virtual population that experienced ALT elevations. This looks very different when the biliary excretion of the metabolite was altered 3-fold or 28-fold, where in the latter scenario, about 1 in 5 individuals of the whole virtual population would have experienced an ALT elevation. And interestingly, even though altering tolvaptan's biliary excretion by itself had very little impact, it appears that altering the metabolite and the parent simultaneously had more than additive effects. So there is some synergy that appears to go on in the case in the bottom 2 rows, since about 1 in 4 individuals experienced ALT elevations when both of these pathways were modified or decreased 28-fold. So, like I mentioned earlier, there's 44 parameters that had inter-individual variability in this virtual population that we used. So performing a susceptibility factor analysis, we identified 5 of these parameters that significantly correlated with the toxicity, which are body mass, CDCA synthesis, and 3 others, which is a nice additional thing you can do with these virtual populations. And this was all in the case when these biliary excretion Vmaxes were altered. So to summarize the DILIsym results, what we saw was that the reduction of this biliary excretion of the parent tolvaptan had very minor impacts, while we saw these very undesirable trends when the biliary excretion of the metabolite was altered. And we saw more toxicity than the additive individual effects when both of these pathways were modified simultaneously. So in addition to the DILIsym modeling, we performed some in vitro studies to support this effort. So we used the C-DILI assay, which is an in vitro assay that basically allows you to test the cholestatic hepatotoxic potential of a test compound, such as tolvaptan. So we used 2 types of systems. We used sandwich cultured human hepatocytes, in which a non-specific MRP2 inhibitor was used called MK571. But we also use HepaRG cells, which are hepatocyte-like, and these are commercially available as wild-type HepaRG cells, as well as MRP2 knockouts. And this latter method with these HepaRG cells, the results are shown here. So triglyceridone is used as a positive control. This so-called sensitization medium basically allows you to really sensitize these hepatocyte-like cells to the cholestatic hepatotoxic potential of this compound. And what you can see is that these MRP2 knockout cells were more sensitive to tolvaptan toxicity. Both the ATP was decreased in this condition, as well as LDH release was increased. So summarizing this DILIsym work and in vitro work combined in this single schematic here, I just want to walk you through the thinking process here. So bile acids that undergo intrahepatic circulation can be taken up by hepatic bile acid uptake transporters and get efflux by the bile salt export pump into bile or back into blood using MRP3, MRP4. So adding tolvaptan to this mix, tolvaptan is metabolized inside the hepatocyte into DM4103. We know tolvaptan is a substrate for PGP and that DM4103 is a very likely substrate for MRP2. In polycystic kidney disease, MRP2 expression and function is impaired, leading to buildup and accumulation of this metabolite, which, well, tolvaptan inhibits BSEP, DM4103, especially when accumulating within the hepatocyte, will inhibit BSEP, MRP3, MRP4, leading to bile acid buildup and hepatotoxicity. In addition, DM4103 is this electron transport chain inhibitor leading to mitochondrial impairment. In addition, bile acids, especially when building up, will reduce ATP synthesis, leading to additional hepatotoxicity. All right, Dr. Watkins, it's back to you.
Okay. Well, thanks, Jim. That's the first publication from use of the DILIsym model by academics, and there's an academic licensing program, and several institutions now have the software. So I'm going back to this diagram I showed you before, which showed that the treatment emergent liver events that were much higher in tolvaptan than in placebo and continued beyond 1 year. And what's shown with the arrows here are the cases that the experts, and I actually chaired the expert hepatology committee that assessed causality. These green arrows are the cases that we thought were probable or highly likely due to tolvaptan. The actual red arrows are the severe so-called Hy's Law cases. And we stopped thinking any of these cases after 1 year were due to drugs simply because it's extremely unusual for a small molecule to produce liver events after 1 year. In retrospect, now that we know what Jim has done, we would definitely say these events, and they clearly are relative to placebo due to drug. The other issue, if you go to the next slide, Jim, this prolonged latency, it doesn't take 5 months to develop a T-cell response, we hope, with COVID vaccine, for instance. And again, I think Jim has provided an explanation of why susceptibility to liver toxicity due to tolvaptan may suddenly emerge over time. And also potentially why when you re-challenge the patient with the drug, it might recur right away. That is, in fact, their susceptibilities just continue to increase. Now, why there would be this very prolonged event is not so clear, but to my knowledge, no one has found any genetic associations with tolvaptan susceptibility, and let alone any immune genetic components. Next slide. So again, I won't belabor this because we're going a little long here, but this is the sweet spot of DILIsym modeling. And I think if you'd asked me 10 years ago, is DILIsym modeling going to be relevant for these delayed, sudden, idiosyncratic liver meltdowns, I would have said no. I think we now clearly know the answer is yes. It's really getting at these early, necessary, but sufficient steps leading up to an immune attack. Next slide, and last slide. So I think in conclusion, QST and in vitro models can provide insights into disease-related mechanisms that contribute to increased DILI risk. Reduced biliary efflux of DM4103, likely due to reduced MRP2 activity, may account for increased susceptibility to tolvaptan-associated hepatotoxicity in some patients. And the modeling was needed here because rats don't get bile acid-induced hepatotoxicity. So you couldn't do these studies even in the polycystic kidney rat and expect to see toxicity. Progression of disease could account for the onset of liver injury after more than 1 year of tolvaptan and potentially the prompt recurrence of toxicity when the drug was reintroduced. So thank you very much, and Jim and I are now available for questions.
Thank you both. That was an excellent presentation. So please put any questions in the chat or in the Q&A. I'll jump in here while we wait. Considering the likely importance of the MRP2 in this work, what future studies could advance our understanding of tolvaptan-associated hepatotoxicity in polycystic kidney disease?
Dr. Watkins, I can probably take that question if you want to.
Go ahead, sure.
Okay. Well, something I've emphasized a few times during this talk is that DM4103 is a likely substrate for MRP2. So we definitely like to know whether it's a true substrate. So performing transporter substrate studies with, for example, MRP2 membrane vesicles and really determining the Km Vmax type parameters would be really useful. And I would so basically we're suspecting MRP2 dysfunction in the patient population as well. There's actually a case study where, like from a few decades ago, where this might indeed be true for some individuals, but really characterizing MRP2 expression or dysfunction at a larger population level would, I think, would definitely be useful to further corroborate some of these findings and conclusions.
Yeah, I can just add to that. I mean, there are liver surgeries done on people with polycystic kidney disease, and an important next step would be actually confirming that MRP2 actually went down. But I think another aspect of this is if, in fact, as seems likely to me, the increased susceptibility in this patient population is due to downregulation of MRP2, what other drugs might these individuals have increased susceptibility to toxicity from? And this is the direction ultimately of QST modeling, is if you understand mechanisms and you begin to characterize larger and larger numbers of drugs and their metabolites in terms of what they do and how they're eliminated, you're going to be able to anticipate these kind of interactions, just as we now routinely do with drug-drug interactions, for instance, but again, at the level of the systems biology, the quantitative systems level. And I think that's the exciting frontier for this whole line of modeling.
Okay, I think we'll have to close there. I know we have one question that came in, but we can follow up and maybe answer that after the presentation. So thank you both again. We really appreciated your presentation.
Thank you.
Thank you for listening. Thank you, everybody.